Hello everyone, welcome to Biochemistry Lesson 1, Glycolysis, and this is the first ever lesson on this YouTube channel. I really hope you guys enjoy this lesson and the content that is to come. And let's get into it. So glycolysis, what is it? Well, it is a metabolic pathway where you begin with glucose and you oxidize glucose through a series of 10 reactions and you eventually get pyruvate or pyruvic acid. And along the way, you also produce 2 ATPs and 2 NADHs. So the two purposes of glycolysis, you could say, are energy production. So you have energy in the form of ATP plus NADH. And the second purpose is to produce pyruvate, which can join the Krebs cycle. And we'll have another lesson on that, which can then produce some energy carriers like NADH or FADH2, which can go to the electron transport chain. And in the electron transport chain, you can produce some ATP, and this ATP can be used by the cell. To do all this, you must first begin with glucose. In glycolysis, you must go through both phases, and you can produce pyruvate, which can be used in the Krebs cycle, and etc. And the first phase of glycolysis is called the preparatory phase because you prepared the molecules you need for the next phase. And also in this phase, you spend 2 ATPs. So you actually use 2 ATPs, so you do not produce any energy here, but instead you use it. So that's why you're preparing the molecules for what's to come. And what's to come is the payoff phase. And in the payoff phase, instead of using ATPs, you produce ATPs. So you produce 4 ATPs. So here we're using 2 ATPs. You produce 4 ATPs, and you produce 2 NADHs. And in net, you have 2 ATPs and 2 NADHs. And this is important that in net you produce overall more ATPs, so that you cannot say that glycolysis uses energy, because it actually eventually it produces energy. And it also produces energy in the form of pyruvate, you could say, because pyruvate has a lot of energy that can be used in the Krebs cycle. And one more thing to note here is that glycolysis has 10 reactions. And reactions 1, 3, and 10 are irreversible, meaning you cannot go back, and therefore they are major regulation points. So they're regulated. The enzymes in these steps are regulated, and the regulation is based on the needs of the cell, the energy needs of the cell, the molecule needs of the cell, and etc. Now we can move on to the first phase of glycolysis and look into it more deeply. Let's begin simply. So firstly, let's say you ate something. So you need to eat something, and when you eat, your body produces insulin. So now you have a higher insulin concentration because the pancreas, so you have a pancreas, right? It secretes insulin, and this insulin goes into the bloodstream. And from the bloodstream, it's absorbed. From the bloodstream, it's uptaken by cells. Not uptaken, but it goes and binds onto receptors on the cells. And let's assume this is the ligand is insulin. And the ligand binds to the receptor, which is an insulin receptor. So this insulin receptor, right, is also known as tyrosine kinase receptors. And tyrosine kinase are a family of receptors which are enzyme-linked receptors, because the intracellular domain of the receptor is basically has enzymes. And the enzyme is a tyrosine kinase because it can phosphorylate tyrosine residues in the intracellular domain. And how this works is insulin binds to the receptor, and it's 2 dimers of heterodimers, because this is a heterodimer because it has 2 alpha and beta subunits, and this is another alpha beta, and when the ligand binds, there's a conformational change, and now those two those two parts can phosphorylate each other. So now you have tyrosine residues that are phosphorylated. So you have 3 on each side, and this can induce a signaling cascade where you have other molecules that bind to this. So now you have this molecule that binds to it, and it can activate something else. It can activate something else, and eventually it can activate these vesicles. And these vesicles have these transporters of glucose. And it goes all the way up here. It joins the plasma membrane. These transporters are now part of it, and now glucose can transport from a high concentration to lower. So it's called GLUT because it's a glucose transporter, and in the liver you have the 4th isomer. There's many isomers, and what this does is it facilitates the movement of glucose from the high concentration. So you have a high concentration here to the low concentration, which is in the cell. And why glucose needs this is because it is quite a bigger molecule, so it cannot just easily pass or diffuse through the plasma membrane. And also it is a polar molecule, so that it's easier for it to just use a transporter and easily go through this hydrophilic interior. And now that glucose is in the cell, it can begin its glycolysis steps. Now the first thing that glucose does when it's in the cell is it becomes phosphorylated. So now you have glucose 6 phosphate, because we added phosphate to the 6th carbon. This is glucose, and this is glucose 6 phosphate. And here we're adding a phosphate group to the 6th carbon. And this is catalyzed by the enzyme known as a hexokinase. So you have a hexokinase, which is a kinase for 6 carbon molecules. And obviously in this step you use 1 ATP. So you hydrolyze an ATP molecule, and you use that energy to add the phosphate group to glucose on the 6th carbon. And this is done for 2 reasons. Firstly, glucose can stay in the cell. Secondly, it makes glucose a target for pathways, including glycolysis. Also, some other pathways can be pentose phosphate pathway, where glucose 6-phosphate is used to produce NADPH and ribose 5-phosphate, or glycogenesis to produce glycogen from glucose monomers, etc. But in this case, glucose 6-phosphate actually goes on to become fructose 6-phosphate, an isomer of glucose, and this is catalyzed by the enzyme isomerase because it catalyzes conversion of one isomer into another. And to be specific, it's phosphoglucoisomerase, and this is a reversible reaction because they are isomers, and this isn't a control point. Really, it's just a reaction which facilitates the conversion of fructose 6-phosphate into the next molecule, which is very important, and that molecule is fructose. However, it's fructose 1,6-bisphosphate. So we're adding another phosphate group, and now it's on the 1st carbon. And you can see here, this was before on the 6th carbon, and now we add it on the 1st carbon. And I forgot to tell you, but the isomers are, so we have glucose 6-phosphate and fructose 6-phosphate. And what happened here was the 1st carbon, which is here, had this bond with the oxygen, but now in the fructose 6-phosphate isomer, you have the oxygen binding to this 2nd carbon instead, right? So then this becomes this, because you add a phosphate group to the 1st carbon, now you have this structure. And fructose 1,6-bisphosphate is very important, and it is catalyzed by the enzyme called phosphofructokinase 1. And make sure to note 1 because there's also phosphofructokinase 2, which is a regulatory enzyme for actually this step. However, that's in the regulation lesson later on. And in this step, obviously, we use an ATP as well. So now we've used 2 ATPs already, as we discussed before. We use 2 ATPs in this phase. and we produce 4 in the next. And now, in the next step, fructose 1,6-bisphosphate has to be broken down into two molecules, and those 2 molecules are, so we have firstly dihydroxy acetone phosphate, and second one is glyceraldehyde 3-phosphate. You can also write it as GAP, and you can see those molecules right here. So this is GAP, glyceraldehyde 3-phosphate. It's an aldehyde, and this is a ketone, an acetone, dihydroxy acetone phosphate. And what you should know is that fructose 1,6-bisphosphate is broken down into those 2 molecules by an enzyme called aldolase. And aldolase cleaves it. So basically we get some form of, so it's this, and here you have a double bond, and this is the cyclic structure of fructose 1,6-bisphosphate. But if you look at it linearly, if it had the linear structure, this would be a ketone here, and here would be an aldehyde right here. So this hydrogen would go right here, and here it would be an aldehyde, and this would be glyceraldehyde 3-phosphate. So fructose 1,6-bisphosphate is cleaved into those 2 molecules, and now what you should know is that glyceraldehyde 3-phosphate is what's used in the next 5 steps. So the next 5 steps begin with glyceraldehyde 3-phosphate, and you eventually get pyruvate. Therefore, DHAP is just useless, right? Well, yeah. The only way it can contribute is that it transforms into J3P, and that is what it does. And the more GAP is used, which the cells do use it, equilibrium favors the conversion of DHAP into J3P. And this is catalyzed by the enzyme triose, because it's a 3-carbon molecule. Triose phosphate has a phosphate group, and it can go reversibly. And triose phosphate isomers, because there are isomers to each other. And then you basically can say is for each glucose molecule, you get 2 glyceraldehyde 3-phosphates. And now, whatever reactions we do in the next 5 steps, we have 2 of each.